Plant kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whitaker 1969, wherein he suggested the five kingdom classification which Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of Monera and Protista having cell wall have now been excluded from Plantae through earlier classifications placed them in the same kingdom. So the cyanobacteria that are also referred to as the blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms and angiosperms under Plantae. Let us also look at classification within angiosperms to understand some of the concerns that influenced the classification system. The earliest system of classification used only gross superficial morphological characters such as habit, color, number, and shape of leaves, etc. They were based mainly on vegetative characters or on the androecium structure system given by Linnaeus. Such systems were artificial, they separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against this, natural classification system developed, which were based on natural affinities among the organism and consider not only the external feature but also internal feature like ultrastructure, anatomy, embryology, and phytochemistry. Such classification for flowering plants were given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on evolutionary relationship between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then proceed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy that uses the chemical constituents of the plant to resolve confusion are also used by taxonomists these days. Algae. Algae are chlorophyll-bearing simple thyroid autotrophic and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stone, soils, and wood. Some of them also occur in association with fungi, lichen, and animals, example on sloth bear. The form and size of algae is highly variable, ranging from colonial form like volvox and the filamentous form like Eulothrix and Spirogyra, figure 3.1. A few of the marine forms such as kelps form massive plant bodies. The algae reproduce by vegetative, asexual and sexual method. Vegetative reproduction is by fragmentation. Each fragments developed into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospores. They are flagellated, motile, and on germination gives rise to new plant. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix or not flagellated non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudrina is termed as angiosperms. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed oogamous, example Volvox and Fucus. Algae are useful to man in a variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy rich compounds which form the basis of the food cycle of all aquatic animals. Many species of Porophyra, Laminaria, and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amount of hydrocolloids water-holding substances. Example algin brown algae and carrageen red algae, which are used commercially. Agar of the one commercial product obtained from Gelidium and Gracilaria are used to grow microbes and in preparation of ice cream and jellies. Chlorella or unicellular algae rich in proteins is used as a food supplement even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae. Chlorophyceae. The member of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to the dominance of pigment chlorophyll A and B. The pigment are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein beside starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by the fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cell, and it may be isogamous, anisogamous, or ogamous. Some commonly found green algae are Chlamydomonas, Volvox, Ulothrix, Spirogyra, and Chara. Figure 3.1a. Phaeophyceae. The members of Phaeophyceae are brown algae are found primarily in marine habitats. They show great variation in size and forms. They range from simple branched filamentous forms, ectocarpus, to profusely branched form as represented by calps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoid, and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of the xanthophyll pigment. Fuchsothin present in them. Food is stored as complex carbohydrates, which may be in the form of laminarine or mannitol. The vegetative cell have a cellulosic wall, usually covered on the outside by a gelatinous coating of algin. The protoplast contains, in addition to plastid, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stripe and leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in moist brown algae is by Bifiliatus zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the oogonium. Oogamous species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common form are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. Rhodophyceae. The members of Rhodophyceae are commonly called red algae because of the predominance of the red pigment or phyophytin in their body. Majority of red algae are marine with greater concentrations found in the warmer areas. They occur in both well-lighted regions close to the surface of water and also at a great depth in oceans where relatively little light penetrates. The red thalli of the most of the red algae are multicellular. Some of them have a complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae as usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gamete. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common member are Polysiphonia, Porphyra, figure 3.1c, Gracilaria, and Geladium. Bryophytes. Bryophytes include the various mosses and liverwood that are found commonly growing in moist shades areas in the hills. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and set localities. They play an important role in plant succession on bare rock soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true root, stem, or leaves. They may possess root-like, leaf-like, or stem-like structures. The main plant body of the bryophyte is haploid. It produces gamete, hence is called a gametophyte. The sex organ in the bryophytes are multicellular. The male sex organ are called antheridium. They produce biflagellate anthrozoids. The female sex organ called archegonium is flask-shaped and produce a single egg. The anthrozoids are released into water where they come in contact with archegonium and Anthrazoid fuses with the egg to produce the zygote. Zygote do not undergo reduction division immediately, they produce a multicellular body called sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division meiosis to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of the little economic importance, but some mosses provide food for herbivorous mammals, birds, and other animals. Species of Sphagnum, a moss, provide peat that have long been used as fuel and as packing material for transshipping of living material because of their capacity to hold water. Mosses, along with lichens, are the first organisms to colonize rock and hence are of great ecological importance. They decompose rock, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. Liverworts. The liverworts grow usually in moist, sandy habitats such as banks on the streams, marshy ground, damp soil, bark of trees, and deep in the wood. The plant body of a liverwort is thyroid, example Markandia. The thallus is dorsiventral and closely appears to the substrate. The leafy member have a teeny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverwort takes place by the fragmentation of a thalli or by the formation of a specialized structure called gamete. Singular gamma. Gamete are green multicellular asexual buds which develop in a small receptacle called gamma cups located in the thalli. The gamete become detached from the parent body and germinate to form new individuals during sexual reproduction. Male and female sex organ are produced either on the same or on different thalli. The sporophyte is differentiated into a foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophyte. Mosses. The predominant stage of the life cycle of moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is a creepy green branched and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organ. Vegetative reproduction in mosses is by the fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organ, antheridia, and archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into sporophyte consisting of a foot, seta, and capsule. The sporophyte in mosses is more elaborate than that in liverwort. The capsule contains spore. Spores are formed after the meiosis. The mosses have an elaborate mechanism of spore dispersal. Common example of mosses are Funaria, Polytrichum, and its sphagnum, figure 3.2. Pteridophytes. The pteridophytes include horsetail and ferns. Pteridophytes are used for medicinal purpose and as soil binders. They are also frequently grown as ornamental. Evolutionarily, they are the first terrestrial plant to possess vascular tissue, xylem and phloem. You shall study more about this tissue in chapter 6. The pteridophytes are found in cool, damp, sandy places, though some may flourish well in sandy soil condition. You may recall that in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in the pteridophytes, the main plant body is a sporophyte which is differentiated into true root, stem, and leaves. Figure 3.3. This organ possesses well differentiated vascular tissue. The leaves in Pteridophyta are small micropyle, as in Selaginella, are large macrophyll, as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendage called sporophyll. In some cases, sporophyll may form distinct compact structure called strobili or cones. Selaginella equisetum. The spongia produces spores by meiosis in spore mother cell. The spores germinate to give rise to inconspicuous small but multicellular free-living, mostly photosynthetic thyroid gametophyte called prothallus. These gametophytes require cool, damp, sandy places to grow. Because of this specific restricted requirement and the need of water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer of anthrozoids. The male gametes released from the antheridia to the mouth of the archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular well-differentiated sporophyte which is the dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are of similar kind. Such plants are called homosporous. Genera like Selaginella and Salvinia which produce two kinds of spores, macro large and micro small spores are known as heterospores. The megaspores and microspores germinate and give rise to female and male gametophyte, respectively. The female gametophyte in these plants are retained on the parent sporophyte for variable periods. The development of the zygote into young embryos take place within the female gametophyte. This event is a precursor to seed habitat, considered an important step in evolution. The radiophytes are further classified into four classes: Psilopsida, Psilotum, Lycopsida, Selaginella, and Lycopodium, Sphenopsida, Equisetum, and Pteropsida, Dreopatris, Patris, and Edientum. Gymnosperm. The gymnosperm, gynomes, naked spermaceti, are plants in which the ovules are not enclosed by any ovary wall and remain exposed. both before and after fertilization. The seeds that developed post fertilization are not covered, i.e., are naked. Gymnosperms include medium sized trees or tall trees and shrubs. One of the gymnosperm, the giant redwood tree sequoia, is one of the tallest tree species. The roots are generally taproot root. In some genera have a fungal association in the form of mycorrhiza pinus, while in some other cycles a small specialized root called coralloid root are associated with nitrogen fixation cyanobacteria. The stems are unbranched cycas or branched pinus cedrus. The leaves may be simple or compound. In cycas, the pinnate leaf persists for a few years. The leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperm are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili bearing microsporophyll and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grain takes place within the microsporangia. The cones bearing megasporophyll with ovule or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be born on the same tree Pinus. However, in cycas, male cones and megasporophyll are born on different trees. The megaspore mother cell is differentiated from one of the three of the nucleus. The nucleus is protected by the envelope and the composite structure called an ovule. An ovule are borne on megasporophyll which may be clustered to form the female cones. The megaspore mother cell divides mutually to form four megaspore. One of the megaspore enclosed within the megasporangia develops into a multinuclear female gametophyte that bear two or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophyte and pteridophytes in gymnosperms, and female and male gametophyte do not have an independent free-living existence. They remain within the sporangia retained on the sporophyte. The pollen grain is released from the microsporangium. They are carried in air current and come in contact with the opening of the ovule borne on megasporophyll. The pollen tube carrying the male gametes grow towards archegonia in the ovule and discharge their content near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and the ovule into seeds. These seeds are not covered. Angiosperms. Unlike the gymnosperms where the ovule are naked, in the angiosperms, the flowering plants, the pollen grains and ovules are developed in the specialized structure called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest wolfia to tall trees of eucalyptus over 100 meter. They provide us with a food, fodder, fuel, medicines, and several other commercially important products. They are divided into two classes, the dicotyledons and the monocotyledons. The dicotyledons are characterized by the seed having two cotyledons, reticulate ventilation in the leaves, and tetramerous and pentamerous flowers, having four or five members in each floral old. The monocotyledonous, on the other hand, are characterized by single cotyledonous seed, parallel ventilation in leaves, and trimerous flowers having three members in each floral hole. The male sex organ in a flower is the stamen. Each stamen consists of a slender filament with an anther at the tip. Within the anther, the pollen mother cell divide by meiosis to produce microspores, which mature into pollen grain. The female sex organ in a flower is the pistil. Pistil consists of a swollen ovary at its base, a long slender. The female sex organ in a flower is the pistil. Pistil consists of a swollen ovary at its base, a long slender style and stigma. Inside the ovary, ovule are present. Generally, in each ovule has a megaspore mother cell that undergoes meiosis to form four haploid megaspores. Three of them degenerate and one divide to form the embryo sac. Each embryo sac has a three-celled egg apparatus, one egg cell and two synergids, three antipodal cell and two polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus, pollen grain. After dispersal from the anthers are carried by wind or various other agencies to the stigma of a pistil, this is termed as pollination. The pollen grains germinate on the stigma and the resulting pollen tubes grow through the tissue of stigma and style and reach the ovule. The pollen tubes enter the embryo sac where two male gametes are discharged. One of the male gametes fuses with the egg cells syngamy to form a zygote. Two other male gamete fuses with the diploid secondary nucleus to produce the triploid primary endosperm nucleus pan. Because of the occurrence of two fusions, i.e. syngamy and triple fusion, this event is termed as double fertilization. An event unique to angiosperm, the zygote developed into an embryo with one or two cotyledons, and the pen develops into endosperm which provide nourishment to the developing embryo. The synergid and antipodals degenerate after fertilization. During this event, the ovule develop into seed and the ovaries develop into fruit. The life cycle of an angiosperm is shown in figure 3.6. Plant life cycle and alternation of generation. In plant, both haploid and diploid cell can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by the plant body by meiosis. These in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexual reproducing plant, there is an alternation of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals represent them differ in the following patterns. First, sporophytic generation is represented only by the one-cell zygote. There are no free-living sporophyte. Meiosis in the zygote result in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant photosynthetic phase in such plant is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as Volvox, Spirogyra and some species of Chlamydomonas represent its pattern. On the other extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by the single to few celled haploid gametophyte. This kind of life cycle is termed as diplontic and algae fucus represent this pattern figure 7.3.7b. In addition, all seed-bearing plant i.e. gymnosperms and angiosperms follow this pattern with some variations, wherein the gametophytic phase is few to multicelled. Third, bryophytes and pteridophytes interestingly exhibit an intermediate condition haplodiplontic. Both phases are multicellular, however, They differ in their dominant phases. A dominant independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte and it alternate with the short-lived multicellular sporophyte totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It alternates with multicellular sporophytic autotrophic independent but short-lived haploid gametophyte. Such a pattern is known as haplodiploidic life cycle. All pteridophytes exhibit this pattern figure 3.7c. Interestingly, while most algal genera are haplontic, some of them, such as Ectocarpus, Polysiphonia, calf, are haplodiploidic. Fucus and alga is diploidic. Summary: Plant kingdom include algae, bryophyte, rhizophyte, gymnosperms, and angiosperms. Algae are chlorophyll-bearing simple thalloid autotrophic and largely aquatic organisms. Depending on the type of pigment possessed and the type of stored food, algae are classified into three classes, namely Chlorophyceae, Phaeophyceae, and Rhodophyceae. Algae usually reproduce vegetatively by the fragmentation, asexually by formation of different type of spores, and sexually by formation of gametes, which may show isogamy, anisogamy, or ogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by the rhizoid. They possess root-like, leaf-like, and stem-like structure. The bryophytes are divided into liverwort and mosses. The plant body of liverwort is thalloid and dorsiventral, whereas mosses have upright, slender axis bearing spirally arranged leaves. The main plant body of a bryophyte is a gamete producing and is called a gametophyte. It bears the male sex organ called antheridia and female sex organ called archegonia. The male and female gametes produced fuse to form zygote, which produces a multicellular body called a sporophyte. It produced haploid spores. The spores germinate to form a gametophyte. In pteridophyte, the main plant is a sporophyte which is differentiated into true root, stem, and leaves. These organ possesses well differentiated vascular tissue. The sporophyte bears sporangia which produce spores. The spores germinate to form a gametophyte which require cool damp places to grow. The gametophyte bear male and female sex organ called antheridia and archegonia respectively. Water is required for transfer of male gamete to the archegonia where zygote is formed after fertilization. The zygote produces a sporophyte. The gymnosperm are the plant in which ovules are not enclosed by any ovary wall. After fertilizations, the seed remain exposed and therefore these plants are called naked seeded plants. The gymnosperm produces microspores and megaspores which are produced in microsporangia and megasporangia borne on the sporophyll. The sporophyll, microsporophyll, and megasporophyll are arranged spirally on axis to form male and female cones, respectively. The pollen grain germinates the pollen tube, releases the male gamete into the ovule, where it fuses with the egg cell in archegonia. Following fertilization, the zygote develops into embryo and the ovule into seeds. In angiosperms, the male sex organ sternum and female sex organ pistil are born in a flower. Each stamen consists of a filament and an anther. The anther produces pollen grain male gametophyte after meiosis. The pistil consists of an ovary enclosing one to many ovules. Within the ovule is the female gametophyte or embryo sac which contain the egg cell. The pollen tube enters the embryo sac where two male gametes are discharged. One male gamete fuses with egg cell, syngamy, and other fuses with diploid secondary nucleus triple fusion. This phenomena of two fusion is called double fertilization and is unique to angiosperms. The angiosperms are divided into two classes, the dicotyledons and the monocotyledons. During the life cycle of any sexually reproducing plant, there is alternation of generations between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant group as well as individuals may show different patterns of life cycle, haplontic, diplontic, and intermediate. Thank you.